Dear students, Namaste. In today's class, we will learn fertilization and implantation in human reproduction. On 14th day, a mature Graafian follicle ruptures to release the female gamete from the ovary. The released cell is a secondary oocyte arrested in metaphase 2 of meiosis, not yet a fully mature ovum. Now let us learn the structure of ovum. Outermost layer of ovum is corona radiata. It is cellular layer composed of rapidly elongated follicular, that is granulosa cells. The cells are glued together by hyaluronic acid and it provides physical protection and nourishment to the egg during ovulation. The middle layer is zona pellucida. It is non-cellular layer. It is thick, transparent, gel-like coat made of glycoproteins. It contains specific sperm-binding receptors. It hardens immediately after the first sperm enters to prevent other sperm from penetrating. Perivitellin space is a fluid-filled space lies between the zona pellucida and the plasma membrane. It degenerates the first polar body expelled during meiosis. Inner layer is plasma membrane. It is cellular membrane having lipoprotein bilayer. Inside the cytoplasm, it contains nucleus, which is haploid. It has 23 chromosomes arrested in metaphase 2 of meiosis. The finger-like fimbriae sweep the oocyte and enters the fallopian tube and rest in the ampulla region on 15th and 16th day of menstrual cycle. During copulation, that is coitus, semen is released by the penis into the vagina. The process is called as insemination. The motile sperm swim rapidly, pass through the cervix, enter into the uterus, and finally reach the ampulla region of the fallopian tube. Fertilization can only occur if the ovum and sperm are transported simultaneously to the ampullary region. When a sperm comes in contact with the zona pellucida layer of the ovum, this contact induces changes in the membrane that block the entry of additional sperms. This mechanism ensures that strictly only one sperm can fertilize an ovum, preventing polyspermy. The secretion of the acrosome help the sperm penetrate through the zona pellucida and the plasma membrane into the cytoplasm of the ovum. Sperm entry induces the ovum to complete its second meiotic division, produces a small second polar body and a mature haploid ootid, that is egg. The haploid nucleus of the sperm fuses with the haploid nucleus of the ovum to form a diploid zygote. The zygote undergoes a series of rapid mitotic divisions called cleavage. The first mitotic division happens about 24 to 30 hours after fertilization, splitting the zygote into 2 cells, then 4 cells, then 8 to 16 cells called morula. As the morula enters the uterus, fluid enters the ball, transforming into a hollow sphere called a blastocyst. Blastocyst organizes into outer single layer of cells called trophoblast and an inner cluster of cells attached to the inside of the trophoblast called inner cell mass. Inner cell mass, each cells are stem cells that will differentiate to form the actual embryo and all its tissues. The trophoblast layer embeds itself into the vascular lining of the uterus, that is endometrium. The uterine cells divide rapidly, growing over and completely covering the embedded blastocysts, establishing a secure pregnancy. Thank you.